Let's dive right into this explainer. Today, we're unpacking a truly fascinating scientific detective story about how tweaking a single specific gene could completely flip our approach to Alzheimer's disease on its head. We are looking at a groundbreaking study that weaves together genetics, the brain's internal immune system, and a highly specific protein-cutting enzyme. We're going to explore exactly how this mechanism might eventually help the brain clear away the toxic buildup that drives the disease.
To really understand this, we're going to walk through the researchers' exact methodology, step by step. Our roadmap looks like this. First, we'll look at the Alzheimer's genetics mystery, followed by testing the mouse model. Then, we hit the plaque pathology paradox, shifting the microglial state, the human genetic connection, and we'll wrap it all up with the unanswered questions.
All right, Section 1, Alzheimer's genetics and the mystery behind it. To kick things off, we really need to understand the biological suspects in this case.
Now, when most of us think about Alzheimer's, we instantly think about neurons, right? The brain cells themselves. But a massive player in this disease is actually the microglia. These are the immune cells of the brain. Think of them as the brain's dedicated cleanup crew. They're constantly patrolling, detecting damaged cells, and engulfing abnormal proteins. This includes that toxic amyloid-beta that clumps together to form those infamous plaques in Alzheimer's patients. Well, recent massive human genetic studies, known as GWAS, pinpointed a specific region of DNA called the MS4A region as a major heavy hitter in Alzheimer's risk. And here's the kicker. Because this specific gene is highly active in microglia, researchers strongly suspected it dictates exactly how these immune cells handle those toxic amyloid plaques.
But science is rarely straightforward. Here is the real problem. Despite knowing there was a strong genetic connection, nobody actually knew if this gene helped or hindered the cleanup process. Did it make the microglia better at their jobs, or did it hold them back? How did it change plaque formation? Which brings us to the core mystery driving this entire explainer. What on earth happens to the brain when you just completely remove this gene?
Moving on to Section 2, testing the mouse model. To solve this genetic puzzle, the researchers needed to see what happens when that gene is essentially flipped to the off position.
So, they set up a functional experiment using mice. First, they started with a group known as 5XFAD mice. These mice are genetically engineered to carry 5 distinct Alzheimer's mutations, meaning they produce incredibly high levels of amyloid-beta and rapidly develop brain plaques. They essentially have the pathology dialed up to 11. Then they took those exact same mice, but completely knocked out the MS4A4A gene. By putting these two distinct states side by side, researchers could effectively throw a biological switch to see if lacking this gene made the amyloid pathology better or if it made it worse.
Before those hard plaques even form, amyloid beta just floats around as a soluble molecule in the fluid surrounding your brain cells. To measure this, the researchers used a super precise technique called in vivo microdialysis, carefully placing a tiny probe into the brain to sample this fluid. What they found was pretty mind-blowing. The knockout mice, the ones missing the gene, had a significantly lower steady state of soluble amyloid and a much shorter half-life. That means the toxic amyloid was vanishing way faster. And crucially, they double-checked and found this wasn't just because the mice were producing less of it. So, early in the disease timeline, removing this gene meant the mice were actively and rapidly power washing the toxic amyloid away before it could even begin to build up.
Now, faster clearance in the brain fluid is absolutely great news, but it naturally sparks the next big question. If this amyloid is getting cleared out rapidly early on, does that actually translate to a reduction in those physical, hard amyloid plaques smothering the brain later in life? This brings us to Section 3, the plaque pathology paradox. Let's zoom in on the physical brain tissue of the older mice to see how this early clearance leads to a really surprising twist regarding the plaque structure. The researchers examined the brains of the older mice using a specific chemical stain called HJ3.4, which highlights absolutely all of the plaque material. As hoped, the knockout mice had a significantly reduced total plaque burden. The individual plaques were much smaller, especially in the hippocampus, which is the brain's main memory center. But here is the paradox. They looked closer and realized there was absolutely no strong reduction in the overall number of plaques. Think about that for a second. The plaques still initiated, but for some reason, the loss of this gene hit the brakes and stopped them from expanding outward.
To figure out why they stopped growing, the team used a second stain called X34. So while that first stain shows the whole messy, sprawling area of a plaque, including all the loosely organized, diffuse amyloid on the edges, X34 specifically binds only to the dense, compact, highly organized core of the plaque. When they compared the two stains side by side, it all made sense. The remaining plaques in the knockout mice had a dramatically higher proportion of that dense core material. Basically, they were far more compact and way less diffuse. It's the difference between a massive, sprawling pile of trash and a highly compacted trash cube. The toxic amyloid was just physically contained much, much better.
Okay, so the plaques are smaller and they are packed down tight. But we have to ask the obvious biological question here. Why? Is this improvement simply happening because a massive army of these microglia cleanup cells suddenly swarmed the area and surrounded the plaques to eat away the edges?
Let's look at Section 4, Shifting the Microglial State. To answer that question, the researchers had to look directly at the microglial cells surrounding those dense plaques using some very specific cellular markers.
They used a marker called IBA1 just to identify the microglia themselves, and another called CD68 to specifically look at the active engulfing parts of the cells. And the results were completely unexpected. They found absolutely zero difference in the volume of microglia near the plaques. No difference in their proximity either. The biological army was literally the exact same size, meaning this incredible plaque containment wasn't happening because they simply recruited more cells to the battle site.
This realization was a total game changer for the investigation. If the sheer numbers are identical, we have to stop looking at the quantity of the cells and completely shift our focus to examining the functional state of the cells. Basically, what on earth were these knockout microglia doing differently?
To find out, they utilized flow cytometry, which lets them look at the markers on thousands of individual cells at incredibly high speeds. And they hit the jackpot. They found that the microglia lacking the gene showed a massive increase in markers like CD86 and CXCR4. What does that mean? It means they had shifted into a pro-inflammatory state. And this illustrates the pivotal turning point of the whole study. Completely removing that gene actively provoked the microglia, forcing them to drastically ramp up their production of an enzyme called MMP9. Now, MMP9 is a protease. It's essentially a set of molecular scissors that chops up amyloid-beta into smaller pieces so it literally can't form those long, dangerous fibrils. All right, Section 5, the human genetic connection. Up to this point, a mouse model is still just a mouse model. The absolute next critical step for the team was to see if this whole MMP9 enzyme mechanism actually holds up when we look at real human genetics.
To verify this, the researchers had to massively scale up their investigation. They didn't just look at a few patients. They analyzed the cerebral spinal fluid, the fluid bathing the brain and spinal cord, alongside the genetic data from 3,506 human individuals. That is a serious data set. They honed in on two specific genetic variants in the human MS4A region. First, they looked at the A allele, which is a known protective variant associated with resilience against Alzheimer's. And guess what? People walking around with this resilient gene variant had significantly higher levels of the MMP9 enzyme in their spinal fluid. Then they looked at the flip side, the G allele, which is a known Alzheimer's risk variant. People with this risky gene had significantly lower levels of MMP9. It was a perfect match. The human data perfectly mirrored the mouse data. A protective genetic state leads to more MMP9 molecular scissors, while a risk state leads to less. This strongly connects this specific enzyme to Alzheimer's resilience in everyday humans.
So let's put the whole puzzle together. Here is the complete, elegant biological pathway that researchers pieced together. Step 1. Altering this genetic switch, whether by complete deficiency in mice or a natural variant in humans, causes a reaction. Step 2. The microglia shift into a specific pro-inflammatory state. Step 3. This new state triggers a massive increase in the production of the MMP9 enzyme. Step 4. Those enzymes act like chemical scissors, resulting in a much faster clearance of the soluble amyloid, which leads to Step 5, ultimately leaving the brain with smaller, highly compact, and safely contained amyloid plaques.
Finally, Section 6, the unanswered questions. Because true science is about rigorously acknowledging what we still don't know. And to their credit, the authors of this paper honestly outline several distinct gaps that desperately need more testing.
For instance, while we know MMP9 increased right alongside the improved clearance, it is still technically a proposed mediator. To prove it's the absolute sole cause of the cleanup, future tests need to treat these knockout mice with an MMP9 inhibitor to see if that protective benefit instantly vanishes. Furthermore, yes, the plaques got smaller and way more compact, but we still need actual behavioral testing. We need to prove that these smaller plaques actually result in improved memory and synaptic health for the living animal. And lastly, remember the mouse model completely deleted the gene, whereas human variants in the real world are much more subtle. So studying human stem cells with those subtle tweaks is the next logical leap.
But even with those gaps, the overarching takeaway from this explainer is incredibly exciting. For decades, we've focused heavily on trying to invent synthetic drugs that scrub plaques away from the outside. But this study suggests a totally different approach. Future therapies might focus on modifying how our own immune cells behave from the inside, literally flipping their genetic switches so they naturally and efficiently clean the brain all on their own.
I want to leave you with one final, pretty provocative thought based on this research. We have known for a long time that chronic brain inflammation can eventually damage and kill neurons. But this exact study shows us that a specific pro-inflammatory state actually helped clear the plaques away. So maybe unlocking the future cure to Alzheimer's isn't about stopping inflammation entirely. Maybe it's about perfectly balancing it, harnessing a targeted, beneficial burst of inflammation to clear the brain without letting it run wild. It is a razor-thin balancing act for sure, but this research gets us one massive step closer to pulling it off.